Welcome everyone to today's explainer. You know, if you've ever stared at a textbook drawing of a cell, it's pretty easy to just picture it as this flat, static bubble. Just a microscopic bowl of alphabet soup, right? But today, we're going to completely change that perspective. We are looking at the cell not as a simple diagram, but as an incredibly dynamic, highly structured architectural marvel. We're going to explore how life essentially upgraded its blueprint to build what is basically the ultimate cellular mansion. And we'll see exactly how unlocking the power of compartmentalization changed the game for biology forever.
Okay, so here's the floor plan for our tour today. First, we'll look at the one-room cell. Then, we'll talk about upgrading to a mansion. Third, we'll dive into the power of the nucleus. Fourth, targeted processing and delivery. Fifth, specialized energy centers. And finally, we'll wrap up with the compartmentalization advantage. So part 1, the one-room cell. Let's look at these simple beginnings.
Let's start with prokaryotes, organisms like bacteria. Think of a prokaryote kind of like a busy, open-concept studio apartment. There are absolutely no walls separating the kitchen from the bedroom. There are no membrane-bound organelles. Because of this, literally everything is happening in one shared space. Processes like transcription and translation, they're totally coupled together, and the proteins that get produced just diffuse freely wherever they happen to be needed in the cytoplasm. But then we have eukaryotes. This is your multi-room mansion. Here, DNA is safely locked away in a nucleus, we've got distinct membrane-bound organelles, and instead of proteins just floating around aimlessly, there's a highly regulated system for intracellular trafficking. It's a completely different level of organization.
So, what do we actually call this multi-room strategy? Well, it's compartmentalization. It's the evolutionary strategy of dividing a eukaryotic cell into specialized membrane-bound organelles to separate and optimize distinct biological processes. And the core problem this solves is basically cellular clutter. See, in addition to the nucleus, eukaryotic cells contain a wide variety of other compartments. By putting physical walls around specific chemical reactions, the cell prevents total chaos. It allows highly complex and, honestly, sometimes completely incompatible functions to happen right next to each other without any interference. It's brilliant.
All right, moving to part 2, upgrading to a mansion. How did we get these organelles? Now, it's important to remember that all modern cells share certain foundational technologies. Like the last universal common ancestor, or LUCA, already had the basic ribosome machinery needed to translate genetic code. But going from that to a complex organism, that took a massive evolutionary leap. Building physical walls and complex compartments required a whole new level of cellular engineering. To get true complexity, life absolutely required those specialized compartments.
Which brings us to part 3, the power of the nucleus, the cell's master control room. Let's walk through the chronological journey of genetic flow here. Because we have a physical barrier, the nuclear envelope, step 1, which is the transcription of DNA into messenger RNA, happens in total isolation. And this isolation is absolutely crucial because it allows for step 2, RNA processing. The cell can safely edit the RNA, splicing out those non-coding regions we call introns. It's only after it's been perfectly processed that we hit step 3, where the RNA is selectively transported out of the nucleus and into the cytoplasm. Finally, in step 4, it finds a ribosome and gets translated into a protein. So this physical barrier of the nucleus, it gives the cell absolute, precise control over when and how a gene is expressed.
Now think back to that prokaryotic studio apartment for a second. Because there's no nucleus, ribosomes just latch onto RNA and start translating it into proteins while the RNA is literally still being transcribed. This coupled transcription and translation creates absolute chaos. I mean, it's a massive, rapid accumulation of proteins. But by putting the DNA in a separate room, the eukaryotic cell safely breaks this chaotic coupling. It stops that rapid amplification from spiraling out of control, which allows for the careful, measured regulation that complex life simply has to have. Okay, part 4, targeted processing and delivery. Think of this as the processing and shipping centers.
So, how does a protein actually get into one of these specialized compartments? Well, let's look at a protein destined for the endoplasmic reticulum, or the ER. Step 1. As a ribosome starts making the protein, a little tag called a signal peptide pops up. Then a signal recognition particle, or SRP, binds to it, briefly pausing synthesis. Step 2. The SRP acts exactly like a microscopic delivery driver, taking the whole complex right to a specific receptor on the ER membrane. Next, in step 3, the protein is threaded or translocated directly across the membrane through a tiny pore into the ER.
And finally, step 4, an enzyme actually snips off that signal tag. Safe delivery complete. The protein is now inside its dedicated workspace rather than just awkwardly floating around in the cytoplasm. And remember, in addition to the nucleus and the ER, we have an entire network of these compartments, things like the Golgi apparatus and lysosomes. Without these specialized membrane-bound trafficking centers, a cell could never properly direct its proteins. Think about it. If you want to secrete a hormone out of the cell to signal a neighboring tissue, or if you need to send highly destructive digestive enzymes safely to a lysosome, you absolutely depend on this targeted delivery system. It is, quite literally, a matter of life and death for the cell to ensure all these materials end up exactly where they belong.
Moving on to part 5, specialized energy centers. Let's check out the cellular power plants.
You know, what's truly mind-blowing about our power plants, the mitochondria and, of course, chloroplasts in plants, is their origin story. According to endosymbiotic theory, these organelles actually evolved from ancient, independent bacteria that literally just moved inside a larger host cell. And now, they're permanently installed generators. Mitochondria power oxidative phosphorylation to make our cellular energy. Chloroplasts provide the perfectly isolated chemical environments needed for photosynthesis. And incredibly, just to show how deeply integrated they've become over time, many essential organelle genes have actually packed up and moved out of the power plant and straight into the host cell's master nucleus.
As the research beautifully states, the relationship between organelles and their host cells has become an obligatory and elaborate one in which no unit can live independently. The host cells simply cannot survive without the massive energy output of the mitochondria, and the mitochondria can no longer survive outside the protective walls of the cellular mansion. It is a perfectly harmonious, completely dependent system. Just incredible.
Finally, part 6, the compartmentalization advantage. Let's really break down why we compartmentalize.
Let's summarize the immense payoff of this system, because there are some major advantages to compartmentalization. First up, precision energy conservation. By controlling transcription and translation across different compartments, eukaryotic cells only express genes exactly when they're needed. They respond to environmental signals without wasting precious resources. Second, advanced gene regulation. By keeping DNA packing and RNA processing safe inside the nucleus, far away from the ribosomes, we get a level of genetic control that's just impossible in a one-room cell. Third, independent microenvironments. Remember, in addition to the nucleus, cells have a huge variety of other compartments. This means the cell can run highly destructive low pH reactions in a lysosome right next to super delicate protein synthesis in the ER, all because they have separate rooms. And finally, cellular differentiation. This advanced regulation is the exact thing that allows a single genome to express completely different traits depending on the cell type.
Which brings me to the thought I really want to leave you with today. We've seen how physical boundaries transform the chaotic one-room cell into this highly regulated, energy-efficient mansion. But think about the sheer scale of what that implies. If a simple one-room cell can only do so much, how did this microscopic architectural upgrade ultimately allow a single, undifferentiated zygote to selectively turn genes on and off, specializing its compartments, to eventually transform into the incredibly complex network of muscle, nerve, and blood cells that make up you? It's a real testament to the fact that, in biology, building walls wasn't about keeping things out. It was about unlocking the potential to build something infinitely more complex. Thank you so much for joining me on this explainer, and keep marveling at the incredible machinery inside you.